Hello, everyone. I'm going to take a little bit of time today to go through sort of a review of the lab, just so we're all on the same page with um, exactly what's being asked with all of the methods and sort of an overview since I've been getting a lot of questions of what's going on. So, um, so I'm going to go through some things. And this video is going to be a little bit of piecewise stuff. So hopefully we can edit it all together and make it okay. But it's going to be kind of raw with a few things that are, um, a little janky, but I'm hoping we get the point across. So the first thing is actually looking at the lab and asking what we're trying to, what's the sort of the application, the use case of what we're doing. And really, it's this idea of converting RNA, which is this four-letter system of ways we encode genes, into amino acids or proteins, right? And we do that in groups of 3 letters. So here on the top of this first image are the RNA bases, GCU, AC, um, ACG, GAG, so on. And if you look, you can use this table on the second image. So GCU, so G on the left is the first row, C is the third column, and then U is the last element within that square. And we know that that corresponds to an A or an alanine, right? Similarly, A, C, G, A is the first row, C is the third column, and G is the first element there, and it corresponds to T, right? So all of this is actually implemented um, in sort of the resources class that I've given you all. So you don't actually have to implement this encoding, but it's good to know how it works. The one thing that might be a little bit tricky is the idea that I have down here at the bottom that some things are stop codons. So as you're encoding these RNA sequences, if you get to one of these stop codons, UGA, UAG, UAA, you stop encoding even if there's more stuff. So if we look at an example of that, so say our sequence was AAA, UGA, GCA. Well, AAA converts into K. With UGA, you're actually going to get back the amino acid, which is an asterisk, right? And GCA corresponds to GCA corresponds to an A. But that means your linked list is just going to be K, right? Because we got to the stop codon and we don't ever view that, right? Okay, we'll see that a little bit later in the actual, once we start creating the amino acid linked list. But that's the basic idea of how that's going. Okay, so let's look at the task that we're given. What we want to do is create a new linked list type amino acid LL, right? Which is actually contains our nodes of our linked list. And if you think about it, a linked list with just one node is actually still a linked list, right? So we might have just one node in our linked list, we might have a null linked list. And it stores these 3 things in a pointer, right? So it stores the amino acid, the codon, and the codon counts. So it's going to look a little bit something like, normally we draw a linked list as a little box with an arrow. So it has the amino acid, the codons, counts, and this is our next. Initially, this is sent to null, right? And it might be null right after it. It could be that our list goes on and on until we get to a null. So this is the null character. Right? But this is our linked list node, right? Is this amino acid LL, right? And we usually have a pointer in from somewhere else. So this is our list. Okay? So I'm going to take some time to go through each one of these methods that I have given the description of here and sort of give an example of what they're supposed to do. Um, I'm going to go through them in the order that they are in the lab description. But you don't necessarily have to implement them in that order. I think the easiest way to implement them is to go from what you think is going to be the most straightforward, right? And the ones that don't rely on other algorithms necessarily being correct or being there for you to test. Um, but you can implement it in whatever order you want to. The base case files that I gave you with almost nothing in them um, should compile and should work. They're going to be wrong, right? But at least you can start testing other things, and I would recommend you doing that. So it doesn't matter what order you do it in as long as it's logical to you. Okay, so the first one is this add codons. So we're given a string, which is a codon, and it will recursively add a codon to a linked list. If the amino acid already exists, it will add the count. Otherwise, it's going to create a new node. So this is pretty much what we use instead of a constructor in this case, right? We're not going to use the constructor from the outside like we normally do. We're going to use this add codon, which is added to the linked list, rather than add it to a specific node element. So let's look at an example real quick, and let's assume that we have these two linked list elements. One stores an A, right, and one stores a C, and they point to each other, and C points to null. And we don't normally label the internal nodes, but just for us to know what's happening, let's label them L1 and L2. And actually the list pointer points to A, so list pointer, right? And we'll go through a couple scenarios of how this works. So the first one is, let's say that we want to add the codon GCU, which is an A, right? So the first thing we call is actually list.addCodon of GCU, right? And list here is actually L1. And it says, is the amino acid represented by GCU equal to A, right? Well, in this case it is, so we say yes. So we're going to increment count of I such that, not such that, let's say where codon I equals GCU, right? So this is, I've written it here as one statement, right? I've written this as one statement, but this is really a loop. You're going to have to go through and find out what index contains GCU, and then figure it out. So what about the case where we want to add something, say we want to call list.addCodon of AGG. So we know, because we have magical powers, that this is actually R, right? So what happens when we do this? Well, the first thing is it gets called on L1, and we ask, is amino acid of AGG equal to A? But in that case, the answer is no. So then it calls on its next, which really should be L2, right? So he says, okay, so that then you call L2 dot add codon AGG, and he asks, or she, is amino acid AGG equal to C? And his answer is also no. Well, there's no L3, right? His pointer is null. So what does he have to do, right? Now we call the constructor. Call constructor and set next equal to new dot 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 dot, right? So basically what happens is each person only knows what their amino acid is, right? L1 only knows that they are A and nothing else. L2 only knows that they are C and that there's a, so they know that they know their amino acid and they know whether or not there's a pointer after them. If there's a pointer after you and you're not that, then you pass the buck down to that guy, right? If there is no next guy, then you know it doesn't exist and it needs to be added to the list, right? So that's what add codon does. Okay, so the next thing is total count. And total count says it returns the number of times the amino acid is used in the sequence. So it's the sum of the codon counts. So here, right, every time a codon representing that amino acid is used, that amino acid is used, right? So total count is basically you're going to return the sum from I equals 0 to count dot length of count I. Right? I'm specifically not writing this in pseudocode, right? Because that's pretty straightforward. But really all it does is it converts, since we don't actually store how many times that amino acid is used, we only store how many times those codons are used, right? We don't need to store both. We can calculate one from the other. So this is basically just calculating one from the other. Okay, so the next one is amino acid compare. So this one is where things start to get tricky, right? So amino acid compare asks us. It says, this is a recursive method that returns the difference in counts between two lists of amino acids, right? So we want to figure out what the difference in counts is. And we're talking about the absolute value of the difference in the total counts. So let's assume that we have list 1, and it's A for C, say 1, R maybe 2, right? And that points to null. And list 2 contains A2, E4, R2, Q1. So notice here that I'm putting a single number, but what these single numbers are is actually the total count values for each thing. So really what we want to do is compare, 1 to 1. So you can sort of assume, right? Like, let's pretend in our head that both of them contained the same list of amino acids, right? We can sort of pretend that there's a C0 in this list, right? And we can pretend that there's an E0 in this list. Everything that's not in the list has a count of 0. And if that was the case, right, if all of the amino acids were in both lists, then we could just go along and compare these, compare these, compare those, right? The difficulty comes in that some of those are missing, right? This guy is not actually here. And this guy is not actually here. So what we're trying to calculate. So before we do that, pretend that they're there, right? And we basically say that A, the difference is the absolute value of 4 minus 2 is 2. C, the difference is the absolute value of 1 minus 0, which is 1. E, the difference is the absolute value of 0 minus 4 is 4, right? R is 2 minus 2, which is 0, and Q is 0 minus 1, is 1, right? So we know for these two lists, assuming that all of the amino acids existed, the difference would be 8, right? But the problem comes in the fact that, like I said, these, imaginary things don't actually exist. But we can process them similar to how we do merge sort, right? If you pretend, so if you start with your pointers, say here, right, at the beginning of both lists, if they match, then you process however you need to process if there's a match, and you move both pointers, right? Now you're here and here. But one thing you know is that C is less than E, right? So I can't tell you, given just the information I know, because remember, all we really know is the information in those two nodes. I can't tell you whether there's an E further down this list. But I can tell you if I got here, there's not, because these are sorted, so the key here is that they're sorted, there's not a C further down in the second list. There could be an E in the first list, but we're guaranteed that there's not a C in the second list. So that means then we can take this pointer and move it here without losing any information, right? We have to process this guy on its own, right, to get this value here, to get this value down here. But we know we can process it and not have to do anything and keep searching. Similarly, when we get to this situation, right, we're going to know that R is greater than E, and we can move this pointer, right, and so on. And then when you get to the pointer pointing to null, then you just basically count out the rest of it. But the key is traversing these two lists in unison, right? And it has to be sorted. So it must be sorted first, right? If it's not sorted, then the traversal, I mean, it could happen, but it's going to be very slow. Okay. Next one is codon compare, right? So codon compare actually uses the same traversal method. So codon compare. So here we were using total count, right? Here, instead of having the total count be the difference, it's going to be, A difference is equal to the sum over the codons of the absolute value of count I minus count other, right? But the actual traversal where you're going along the line, the lists stays exactly the same. The only thing that changes is the way you calculate difference, and I gave you the differences here, right? But the traversal stays exactly the same. So the next two are basically our getters, public char array, amino acid list, and amino acid count. Here, I'm gonna actually describe them together. So amino acid list, amino acid. So these are getters, but they're going to be recursive, right? So again, assume that we have this list. Call it list A4, C1, R2, right? So that same list one from above. This guy points to null, that guy points there, that guy points there, right? So list dot amino acid list is going to basically return A followed by list dot next dot amino acid list, right? We know, because we're smart human beings, that it's actually going to return A, then C, then R, right? The problem is that this node here, this A node, does not know how long this list is going to be. So a little hint here is that R knows how long that list is going to be from him on, because he's the last node, right? But these other guys don't know how long they're going to be until they get the result back. And then lists dot amino acid count is going to return 4 and the same thing, right? List dot next, blah, 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 blah. And we know, again, because we're smart humans, that it's 4, 1, 2. But just like up above, A doesn't know how long the rest of the list is. So it's going to have to be recursive. Right? So the next guy is sorted. So this is another sort of getter, right? And this is pretty straightforward, but it tells you is sorted. Is the list sorted? So is the set of amino acids, are they in the correct order? And we can use ideas from bubble sort here, right? Where, remember, if you watch the bubble sort video or if you read it in the book, you can short circuit bubble sort kind of, right? Where if you make a pass, so we had A, C, R was our example, right? If you make a pass and you look at every pair, if you look at every pair and they're in the right order, then the whole list has to be in the right order. If you ever make a pass and one of the things is out of order, then the whole list is out of order or is not in order, right? So you may, if you think again, this A only knows about himself and the one guy after him. But if those two are in the right order and everything else beyond them is in the right order, then the rest of the list is in the right order. And this last guy, R, well, he's a list of one. Is that list of one sorted? Yeah, a list of one thing's always sorted. So that's how is sorted is supposed to work. So create from RNA sequence takes an RNA sequence and returns the linked list containing only the amino acids present in the sequence with the codon counts propagated. Note that the stop codon does not actually exist in the list, but indicates when translation should stop. So the way this is going to work, and notice that these two methods, create from RNA sequence and sort, are both static, right? They actually build something not on an instance, but in general, right? So they're working without any concept. All of the previous methods that we had talked about were on a specific instance. Now we're talking about something that works sort of in general, right? So create from RNA sequence. Assume we're given some string, so it's given a string S. Let's say that string S is AAA, CUG, right? Initially, we start with our list is null, and we process our first codon as K, right? This guy is K. So then our list ends up with K, and that points to null. And once we have our first element, then we can run, say this is L, right? Then from then on, we can run L.add codon. Until what? Until either we reach a stop codon or reach the end of S, right? S is our string. So one of those two things is going to happen, and when that happens, then we return, right? Then you return L. Because now L contains all of the amino acids. If you've implemented the constructor right, and add codon right, then all of those codon counts should be correct. Right. The last thing is sorting. So sort, it takes a list and returns a list. Why does it need to take and return a list? Well, that's because if we originally start with, say, Z, A, F, right? L in, L out, right? L in points to Z, where L out points to A, right? Because the list is going to end up being A, and then this pointer points to F, and this guy points there, and that points to null. So you can use any sorting algorithm you want, right? We've talked about 4 in class. We've talked about insertion sort. We've talked about selection sort. We've talked about bubble sort. We've talked about merge sort. any one of those you feel most comfortable with is the one you should use. I'm not going to specify which one I want you to use. It's up to you. Okay. All right. Well, hopefully all of this helped. If you still have more questions, please feel free to let me know. I'll be around um, most of the rest of the week and, over the weekend and all that fun stuff. Okay.